So the caption says, if this doesn't help you, don't worry, it's a placebo. You get into an interesting ethical question with things like this, is that, because placebos, for things that we have some mental control over, how much we feel pain, you can actually control certain things like headaches and such to a certain extent, then placebos are effective treatments that are no treatment at all. And you come down to the question of, take acupuncture for instance. Is that acupuncture is very powerful placebo effect. How do we know this? Well, they've tested it a bunch of different ways, some of which are not that satisfying, but a couple of which are very, very good. Acupuncture is the idea of putting needles in particular places, invented by the Chinese government based on sort of something that they did going back a ways. But just give you a little history on the ancient Chinese medicine. There was a purge. The communist government in China executed a purge to the late 50s and early 60s that took out a huge number of educated people, including most of the Western trained doctors. And then they had a big country and no doctors. So they put together an organization to kind of dip into different aspects of Chinese folk medicine, reconfigure them in ways that they could use and that they could train people to do really, really quickly. And then just kind of threw them out there so that people had options for, if they hurt, they could go see a doctor. And at some point, it got picked up in other cultures and became this ancient Chinese medicine. And since it was a marketable concept with a lot of products, is the government kind of they rolled with it. It's like, sure, we'll sell you acupuncture needles and all this other stuff. But at least partly an invention of necessity, while they raced to train doctors in actual evidence-based Western techniques, which, as I understand it, and I don't live in China, haven't lived in China, but as I understand it, your typical educated Chinese person is going to use Western medicine and not ancient Chinese medicine, because they kind of were on the ground when this all developed, or at least heard from people that were. Anyway, acupuncture in its official form involves putting needles in, I think they're called meridians, particular spots on the body that are supposed to affect flows of qi and other kinds of things that are not measurable in any kind of way, and redirects and all this other stuff. So some of the earliest tests of acupuncture used needles, but they just had people put the needles randomly. They didn't put them in the right places. And it turns out if you're treating your control group, getting random positions, getting your experimental group, getting them the correct positions. For a lot of things that we do have control over, experiences of pain, experiences of just kind of general malaise and illness, things like headaches, they got better. Both groups got better, and it was a very powerful effect. Laying on a table, have people stick needles in you, produces a very powerful treatment placebo effect. Later on, somebody invented a type of placebo needle based on stage daggers. You're on stage and somebody stabs you with a dagger, and it goes in because the blade goes into the handle. And so it looks like you've been stabbed, but obviously you haven't been stabbed. Is that they developed a fake acupuncture needle that was a little narrower on the bottom and had a shaft that could go up. And then before the treatment, the edge of the bottom of the shaft was sticky. So what they could do was take the needle, you could watch them, they'd put it on the proper spot on your skin, and look like they were pressing it in. Now you could feel the tip of the needle, so it felt like they were pressing it in, you could see it go. And then it would get to the base of the shaft and stand there, because it was sticky. And so it looked like you had a whole bunch of needles in you. Very brilliant control system, right? You want it to be as much of a match to the real thing as possible. Still, placebo group, non-placebo group, same effects, but the thing is you do get effects. And so some people go, well, for folks that have chronic pain or they have some sort of problem that Western medicine really can't address, is it really ethically wrong to send them to acupuncturists where they kind of use the power of their own perceptions, their own expectations to make themselves feel better? And that's a tricky question, is it because the flip side of that is, well, yeah, but now you're sending them to one thing that really isn't based in evidence. What they claim is happening isn't really what's happening. And how much do you open the door for everything else that's out there that in some cases is not working at all, in other cases is actually actively harmful? So there's a question. Good old ethics. We got a little bit more of the chapter to do. Reproducibility. You do your study and you have your materials and methods, your process. And if somebody would get all of that and duplicate your experiment, they should get roughly the same results. They're probably not going to get exactly the same results, but they should get roughly the same results, or something's a little screwy. And reproducibility is tricky because people don't reproduce other people's experiments except under two cases. They think there's something wrong, or they need to reproduce it because they're going to take their experiment and build on it, is take it in a different direction. Like the guy with the aluminum, is that he wanted to see, on the assumption that that the reproduction of the first step from the original test would give him aluminum infused brains, he wanted to see if he couldn't figure out where exactly the aluminum was and what exactly the aluminum was doing. And then he found out he couldn't reproduce the results in the first place. There's, over the last few years, been what's called a reproducibility crisis in a number of different sciences where people do that. They start with what's supposed to work, and a lot of times it doesn't work. Pharmaceuticals do that. I remember I said academics tend to find a lot of basic stuff. That's what's happening in the cells, is what's going on here. Well, the pharmaceutical community follows that stuff because they'll see a new metabolic pathway or a new chemical reaction inside a nucleus and go, oh, okay, we can possibly connect that to a condition, and now that we know how it works, we may be able to come up with a treatment for it. And a lot of times, they can't reproduce the original results to, their first step, it just goes nowhere. And that's actually been a kind of a dirty little secret in pharmaceuticals for a long time. But in the last couple of years, people looking at even things like cancer treatments, psychological stuff, big time, they found that a huge fraction of tests that weren't really reproducible. And they ascribe part of it to the rush to get stuff published. Publication is your point system in science, is that the more you publish, the more people use the stuff that you publish, the higher your status. Sometimes that's what you need in order to get promoted, or at a university to get tenure and to stay there, to get funding for grants. So there's a huge incentive to do it fast, get it printed out, and put it out there. And so there's also an incentive to cut corners and maybe fudge a little bit. And so there's issues with that. It's science, it's done by people. People have motives and temptations the same way as anybody else. But at its best, your study should be reproducible under other circumstances. And if it's not, either there's something about your study that's iffy, something about the reproduction that's iffy, or you find out weird things like your study doesn't work everywhere. People did a reproducibility study on mouse model studies a number of years ago in different locations. but they had like this traveling kit. So they were able to go from New York to Denver to San Francisco to other places and set up in a lab and run a couple of basic mouse experiments in those different places. And they found they got different results. And they didn't really know why. Is that in some cases like Denver, there's altitude, but in other places it's like, why are the mice behaving badly, or not badly, but differently in other places? And that got published, and people grumbled about it for about 5 minutes, and then everybody just kind of, apparently forgot about it. Because what do you do? If you're doing your preliminary experiments with mouse models, and here's a study saying that they're going to be different in different places, you don't really have a viable other option except to kind of ignore that and hope that it's wrong. I know, I'm making science look terrible. But at its best, it's a great system. It works really well. It gives you evidence to support whatever it is you want to do. That's what we're in the middle of right now, is that we're in a pandemic where we're trying to make decisions based on what we've tested and what we've seen done, even though a lot of times we're kind of making it up as we go along and then kind of looking back to see whether it worked okay. When you designed your experiment, you decided going in what it was that you wanted to actually measure to see the effect that you were expecting. You're right, predictive. If we do this, this should happen. How do you know that? Well, we'll measure this or this. Sometimes what you also wind up doing is saying, okay, I'm gonna measure this. This is what I think the really good measurement is. But I've got the equipment and it's set up so that I can measure this, 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 and this also. And hopefully I'll get supporting evidence from these other measurements. And sometimes you don't. You measure a couple of different things and one of them contradicts the other. Or if you measure a few different things, going in, you have to think in terms of, so what do I do if the measurements don't jive with each other? Is it, do I go with my main thrust and kind of ignore the stuff that doesn't support? Do I factor it in somehow? It gets very, very tricky when you design an experiment. And for us, when we do our simple experiments in the lab, just measure one thing, just test one variable and measure one thing. Because if you measure A and A is great and B sucks, what do you do with that? You should know that going in if you're going to design your experiment that way. But your results should support your hypothesis. And sometimes what happens is you have your hypothesis, this should happen. You have your null hypothesis, this should happen if it's not right. The very first landers we put on Mars were supposed to do chemical tests on the soil. And they said, well, if this happens, it means that there's been life in that soil. If this happens, it means there hasn't been life in that soil. So they were looking for A or B, and of course they got C. They got chemical results that were like, what the heck does that mean, sort of results. Interpreting results. Since this is a fairly short part of the chapter, I'm going to give you another good experiment. I used to be part of the Great Lakes Research Consortium. You go to conferences and listen to people's research, and they tell you something. PCBs are chemicals produced by a number of different processes, but for a long time GE produced PCBs as a waste chemical when they made insulators. GE big into turbines and electricity and stuff. And the PCBs were these big, supposedly inert compounds. So they were kind of flushed out into the water, and they being heavy, big, heavy molecules, they sank into the sediments. But at some point, tests indicated that that they could be carcinogenic, especially some of the breakdown products could be carcinogenic. So the question then became, so, are they, what else can they do? This is what one of the researchers decided. She wanted to see, since PCBs are lipid soluble, they can get into your cells and they can accumulate in fatty tissue. That's why if you're a fisherman in New York, if you read the instructions, you're supposed to scrape all the fat off the fish when you catch them, because there's PCBs basically in any waterway in New York. PCBs also were thrown into the atmosphere and have settled in a lot of Adirondack lakes. Anyway, Here was her experiment. She wanted to see the effects of PCBs on behavior. So she used rats. Rats are a very good behavioral model because they're pretty smart animals and people have been using them for a long time. So we've got a lot of model setups to test this sort of thing. The test was essentially a test of persistence. Here's the test itself. You've got a rat in a container, and at one side of the container is a little food dispenser with a little release on it, a little thing that hangs down that they can press with their feet and get a release, get food. But it's set up so every time you get food, the next time to get it out, you have to hit the bar, excuse me, twice as many times. Hit it once, get food. Hit it twice, get food. Hit it 4 times food, 8 times food, 16 times food, on and on and on and on. And what you do is you set up a graduate student. Now we probably do it on video, but they didn't have that much video then. Graduate student takes your nice little friendly rat, puts it in the container, starts the clock, and it's like, okay, what are we measuring? Grad student, when are you stopping the clock? And they say, okay, we're gonna measure this until the rat essentially gives up. Okay, how do you define gives up? They move away from the bar for at least 5 minutes straight. You can stop the clock then, and we're gonna talk about how long they're persisting at trying to get the food. And what they did was they fed the experimental group salmon caught on the East Coast, where the salmon do have PCBs in their tissues. And they fed the controls Pacific salmon, no PCB contamination out there. So they gave them the same food, and the difference was the variable, PCBs, in the food for the experimental group but not for the control group. So, boom, food, bum, bum, food, bum, 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 food. Here's what they found. The PCB, the experimental group, wouldn't give up. They went so long they had to swap out observers. Is that they were doing things nobody had ever seen with this type of setup before. Is that they actually would get to the point where they needed to actually hit the bar a couple of 1,000 times to get the next food. They would come over and kind of loosen up their front legs and get on the bar and just go and they would just do it until food came out. They'd eat the food, they'd back off for a couple of minutes, then they'd go back and go at it again. That's your result. She's in front of this conference of people, and she looks at us and she goes, what does this mean? You know, this is an animal model of human behavior, but I don't know what these results, they would not stop. I don't know why. And I don't really know whether I'm really measuring persistence or some sort of psychosis that the rats are having. But that's happens sometimes. You get results that you totally did not expect, and now you have to interpret them. You interpret your results. It's weird that we are an animal that tends to think in numbers. Because persistence is just, it's a subjective thing, how persistent. More persistent, less persistent. But we want to go, well, we have to come up with a way to measure it. We have to come up with a way to convert that to numbers. And we do that all the time, is that pain is a continuous scale. It's a qualitative feature, but we, in order to measure it for experimentation, measure it in numbers. And we do this in ways you're not even thinking of. Time is a qualitative feature. I'm pointing at the clock. Is a qualitative feature. It's a continuous thing, and yet we split it up into hours and minutes and seconds and don't even think twice about that's an artificial construct. Same thing for temperature and other things about the world that we communicate with, and that's one of the strengths of quantitative, is that quantitative, converting things to numbers, makes it understandable when we communicate to each other. You go, oh, it was 36 degrees yesterday, and somebody from the United States goes, isn't it summer? You go, oh yeah, that's Celsius. And so, you got to convert it into a number that somebody can understand. But one of the strengths of quantitative is that. The other thing is it gives you measurables. It gives you ways to do your experiments and have a measurement for how long did it take before they stepped off for 5 minutes. Click. I can compare that to this rat, to this rat, to the control rats. I can average those numbers. All of that kind of manipulation is something you really can only do with quantitative results. And of course, that brings us to statistics, which is how do you manipulate the numbers to get the answer that you're hoping to get? And that's the problem, is that there's hundreds of different types of statistical manipulations. There are so many now that when an old guy like me took statistics, we learned the math. Now, as I understand it, when you take a statistics class, they basically show you how the software works and don't really focus that much on what the software is doing mathematically. This, here's a statistical program that'll do this, and here's one that'll do that, and here's one that'll do that, and here's how you feed the data in, and here's what the results mean. And they, as people have told me, they don't really go into the math, which is just as good, because my statistics instructor, now that I'm thinking about it, now that I know how the system works, my statistics instructor was probably a graduate student in math who was assigned to do statistics and wasn't really that much of an expert on it. Because I'd like to understand how things work, and I would go up after class and go, okay, I understand this part and this part and this part of the equation, but at the end we add 2, plus 2. I go, why? I don't understand what the 2 represents. Why do we put 2 at the end? And he goes, because it works. That was his answer for, I'm sure somebody with a keen statistical mind could probably have told me why the 2 was there. But it does get very, very complex. It's so much so that there's an old expression. There's lies, there's damn lies, and then there's statistics that kind of to say you can make the numbers dance the way that they dance. And it's useful to understand the math. This is the middle of August, last Sunday, or the Sunday before, was the big deal where the guy from Axios had interviewed President Trump, and he had all his papers. And to start off, he's bragging about how we do more testing. We've done more COVID testing than anywhere else in the world. And the main reason for that is we now are doing a lot of testing for people for their jobs, for people in nursing homes or people in prisons, and so there's a lot of repeat testing. This many testing is not necessarily this many people. But we got lots and lots and lots and lots of tests, which gives us lots and lots and lots and lots of positives. And then he hands a statistic over, and it's the death rate. And he goes, well, that's, we're better than some of these other countries. That is the death rate measured against the number of tests. So the number of deaths against many, 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 many tests, positive or negative, is going to be comparatively a lower number. It's going to be a smaller number compared to this really big number. Whereas if it was, say, the deaths compared to the number of positive results, then which is the rate that everybody else uses, then it's not so nice a number. In Fulton County, I was surprised to find that the rate of death compared to the rate of positive results is almost 1/10. So somebody who gets a positive COVID result in Fulton County has, at least did up until a couple weeks ago, I haven't looked at the data lately, has like a 1 in 10 chance of dying. Way higher than almost any other place in the state, including, Montgomery County is way lower than that. But anyway, statistics, rates. Remember, convert stuff to numbers. Numbers make more sense to people, but you got to be careful. Is that we used to have a lab on campus that everybody in the lab was comparing things in coyotes. And every time one of the graduate students, the many, many graduate students who worked in that lab, I was the only graduate student in my lab because it's invertebrates, who cares? But they would come out, and every single time they'd show us essentially the same stuff, but their results would all have been filtered, each one filtered it through a different statistical approach. And it really gives you the feeling of, let's run these numbers 12 different ways, and whichever one looks the best for what we want it to look like, that's what we're gonna choose. And so that's one of the downsides of using statistics. And then, of course, this brings you to conclusions. Here's our results. This is what the results mean. When I've gone to conferences on a number of occasions, now partially this is because I'm not necessarily a peer to the people that are giving these talks, so there are things I don't totally understand. Odds are if my conclusions differ from their conclusions, I'm more likely to be wrong than they are. But that happens where they're walking their way through and they go, here are our results, here are our results. And you're sitting there going, oh, okay, I think I see what's happening. And then they go, this is what we decided was happening. And you go, really? And me being me, I go, what about this? And then most of the time they'll go, oh no, you're thinking of this and this is why. And they kind of take my brain and put it in a better spot because they understand it better than I do. But also there are times where I just out and out disagree. You look at somebody's results and totally disagree with what they concluded from their results. And I wish I had a good example of that, but I don't. I'm a storyteller and I expect the examples to be in my head when I need them rather than write them down, and they're not there right now. You'll know that. The metacognition lab will cover that. Okay, that is, what is it? That is all of chapter 3. You will get a chance to really experience this better in the experimental design lab. I'm trying to decide whether that's going to be one of the remotes. I'd rather it was one of the participation labs, but it isn't really hands-on, it's brains-on. And so I may wind up having to do it differently.